There's been a lot of talk about MSG and aspartame as being harmful substances, or in some cases there's a defense of them as being harmless substances. But in this video what we're going to do is look at what happens to those substances in the body, how they are digested. Okay. Now, before we get into the basics of that, we're going to look at some background material which relates to amino acids and proteins. Okay. So, proteins are essentially a string of amino acids. Okay. Think of it like a chain. All right. You have a chain with many different links, okay, and they're all hooked together. That chain is like your protein, okay. Each link is the amino acid, okay. So the amino acids are like chain links that strung together form proteins, okay. But let's look at this amino acid from a chemical perspective. It's an amino and it's an acid. Okay, it's an organic compound, of course, and organic compounds have what we call a carbon skeleton, typically, and then some functional groups. Okay. An amino acid, this is telling us what functional groups that organic compound has. It has an amino group and an acid group, okay? And they're in a particular arrangement, okay? So an amino group looks something like this, okay? It has that nitrogen with two hydrogens attached to it. Many times, depending on how it's written, you'll see it on the other side, NH2. It's the same thing. Right? And then the acid group, it has a carbon, and attached to that carbon is a double bond to oxygen and a single bond to another oxygen that has hydrogen attached. Okay? So now for simplicity, we often don't write in the carbon atoms with their letters. We just kind of imply them with this line drawing notation. I have a video that explains how that works. But here we have represented the amino group, all right, and the acid group. Okay, so every amino acid has those two groups in that arrangement. And the thing that makes them different is that attached to this carbon in the middle is, I'll just write a generic R group, okay, where R represents quite a number of different things. There are actually, well, there's 20 common amino acids that are typically encountered, and we'll talk about that a little bit later. But the only thing that differs is what is attached at this middle carbon. Okay. Now, it also happens that there's a particular three-dimensional arrangement between these groups that are attached at this carbon. I'm not going to go into detail about that, but the proteins only have one particular arrangement. It is specific with that. Okay, so this is the structure of an amino acid, and if you take say another amino acid and you put it side by side with one that we have there, okay, there's our second amino acid. All right, now how are these chain links hooked together? Well, you'll notice here we have OH and we have some hydrogen over there. If we take those, all right, HHO, that's water, okay? So essentially, if we just remove water, we'll hook that nitrogen up to that acid carbon there, or the carbonyl carbon as we call it. Right? So what you end up with is you get rid of this water in what we would call a condensation reaction, because you're forming water, right? And you end up with a combination of these two amino acids. So there's the one, we've gotten rid of the OH, and now we have just one hydrogen left, but it attaches at the carbon, or rather at the nitrogen, and then we have, and I'll go ahead and just draw it this way, flip it around a little bit. All these bonds, they rotate on these bonds, so it's not terribly important how I draw that. But here we have now the combined amino acids, just losing that water molecule. Okay, so that's what happens. One after another comes next to it, loses a water molecule, and gradually the protein is built. Of course, this is done in the body with the help of enzymes and whatnot. But here we have what is called a peptide bond. Okay, the bond that hooks those two there, that forms the peptide bond, right? And we call this the peptide backbone. This would be the peptide backbone, actually not including the hydrogen or the OH there, because those are the linking elements. But there's our backbone, the peptide or the polypeptide backbone. And then we have these different R groups. Okay, that's from the different amino acids, and the protein may have any number of different amino acids as different R groups on that peptide backbone. But the backbone itself is going to be the same in any protein. All right, so that's how proteins are constructed from the constituent amino acids. All right. So what happens when you eat protein in your meal, you have it in this long chain. All right. Here I've only drawn two, but protein has hundreds even of these amino acids chained together, and so it is indeed a long chain, depending on the protein. And what happens is, in the body, it breaks them down. Okay, it's a process that is just the opposite of what is done here to form those bonds. If you take that water and you insert it at that peptide bond, what happens is it breaks it. Okay, it's a process that is called hydrolysis. Okay, two parts to this word: hydro, referring to the water, and lysis, referring to the splitting apart or the breaking of that peptide bond. Okay, so hydrolysis is just the process of taking it back from the protein to the individual amino acids. Okay, the opposite of condensation. Okay, so we'll come back to that in a little bit, but that gives you just a brief background of proteins and their relation to amino acids. Okay, now let's